Hello, good morning, friends. I welcome you all in the refresher course sponsored by UGC HRDC Gujarat University. I welcome you all for this refresher course. Myself, Dr. Vikram Rawal, am working as an assistant professor, Department of Microbiology and Biotechnology, Gujarat University. I'll be taking the topic that is role of biology in the forensic science. Now, what is forensic biology? Whenever any science is used for the investigation of any criminal offenses and which can help out the judiciary system to solve a case, the science then becomes a forensic science. When biological theories are applied to the forensics, it is considered to be forensic biology. In general, the biology is used to associate person or persons, whether they are suspect or victim, to a location, any item or a group of items, another person which may be again victim or suspect respectively. There are few basic principles upon which the entire science of forensic investigation depends. Now we'll see one by one each of these laws and principles that are used for forensic studies. Now these are the list of various principles and law that we take into consideration for studying or solving anything that is related to forensic science. The first come first is the individuality principle. Next is the exchange principle. Third is the law of progressive change. Then the law of comparison. Next is the law of analysis. Then comes the law of circumstantial facts. And lastly, the law of probability. These are the laws that are playing a vital role in forensic studies and all of the forensic investigations. First, the individuality principle. The principle of individuality as is being attributed to Paul Kirk in 1963, and he is regarded as a building block for forensic science to attain this huge stature in various fields of science. Individuality implies that every entity, whether a person or an object, can only be identical with itself, and it is so unique. So anything that we consider, it is unique in that aspect that is similar to itself, nothing else. It cannot show any kind of similarity, be it 100% with any other thing. If it is 100%, it is said to be matching with itself and no other thing in the world. No two objects, whether they are natural or artificial, can be exactly the same. Kirk also claimed that the main aim of forensic science is to focus on the source of two items, questioned and known, or mark and the print, which are thought to have come from a single source. As such, identification is also concerned with establishing individuality from traces that are left at a crime scene, rather than the two things being exactly the same. This means that identification can be shown indirectly through the analysis of various traces and samples. Again, we say no two fingerprints in the world are same. It can marginally match with one another, but in a world, if a person is having fingerprint, it is considered on the individuality principle to be very, very, very unique. Next, the exchange principle. The exchange principle is attributed to Edmond Locard. The principle states that whenever two objects or subjects they interact, some sort of traces will be left behind. This is generally what happens at any crime scene. Trace materials include hairs, blood, fibers, as well as gunshot residues. Locard suggested that there are many traces which are left behind, and if these are interpreted properly, they provide the most valuable information to solve any of the crime scene. Now I'll take example of my two hands. If my two hands are rubbed with each other, there will be friction, there will be some sort of scuffle that is taking place. Fine. And very minute amount of cells which are lying on the surface of skin, these are the dead cells of my skin. If I rub it continuously, there will be these minute cells which will be withering off from both the hands, and these traces will be left onto either the other hand, or they will fall at the crime scene, or they will fall on the clothes likewise. Fine. So it is said that whenever there is a hustle between the two, there is the suspect and the victim, there is a hustle, so there might be something, that might be some traces which may lead to a very good lead in the forensic investigation. The third is law of progressive change. Now when we talk about biology, biology is a science in which all of the biological material, they tend to change their phase. They change their the structure, they change the chemical compound. If whatever biochemical compounds are present, these compounds may change, they may deteriorate, they may degrade in due course of time. The exposure of these compounds to the environment may eventually lead them to be degraded, distorted, and it may not be the exactly same as it was on day one, and when it is taken into consideration for analysis after a few days, when the actual forensic investigation team comes, or when the sample arrives for the forensic analysis. So here what we say is the law of progressive change. So different objects, they change, although they may change across different time spans. All of the compounds, they not necessarily change at the same speed, at the same rate. If it is of biological origin, if it is a live cell, if it is a body fluid, it may change in due course of time. It will be having few days lifespan. But if it is something that is of non-biological origin, it may degrade, it may deteriorate, it may have structural changes, but it may not totally be decomposed. If it is a biological material, a few time lapse may eventually lead to a total decomposition of that biological material. Now for example, we say blood samples which eventually degrade. The blood sample which is actually found at the crime scene, fine, may not be, if it is not analyzed at that very juncture, and if it is collected and if it is sent to the forensic analysis, it may not be that immediate. Fine. So in due course of time, whatever sample that was taken, it may degrade, it may deteriorate, and the actual analysis may be affected. I am not saying it is totally affected, but it may be affected. Some objects are more durable than the others and may be relatively said to be permanent in nature, remaining mostly unchanged during their identification. Say for example, if a metal weapon is taken. Now this metal weapon, in due course of time, it may, if it is exposed to moisture, if it is exposed to many other things, it may get corrode and even some rust may be formed, but it may not totally degrade. Fine, the article remains the same. If it is a knife, it is a sword, if it is a hammer, anything, it may not totally degrade. If it is of biological origin, yes. If it is of biological origin, 100% in due course of time, the material is bound to deteriorate, material is bound to degrade. So if an object is very durable, it may quite be easy for its use for identification. If it is less permanent, its main features, they change. During the identification process, it is not possible to answer the question of that thing being same or the original one. Fine. So often it happens that it may, if it is not analyzed properly within due course of time, it may eventually lead that we may require some fresh sample. And what if the person is already deceased? If a person is already dead, you cannot have its blood, you cannot have its other body fluids which are to be analyzed in certain cases. Right? So there is a law of progressive change, and it strictly applies to all of the biological materials. Next is the law of comparison. Different samples must only be compared to the samples which are alike. I cannot compare a dog and a giraffe, right? So it is said that the samples which are obtained must be analyzed and compared to samples which are similarly looking or samples which are look-alike. I can compare, if I am comparing blood, I should compare blood samples with the other blood samples. There is no point in comparing blood samples with other, like sweat sample or semen sample or any other XYZ body fluids. Right? So in other words, blood samples are compared to blood. Similarly, if it is fiber, if it is a cloth, it might be compared to cloth, fine, so on and so forth. If it is a weapon, it might be compared to weapon only. If it is a gunshot, whatever these bullets, whatever residues are taken, they must be compared with that only. Right? The sole identity and individuality of any sample can only be established after a thorough comparison with all the possible lookalikes of that sample. So that is law of comparison. Next comes the law of analysis. The quality of analysis is determined by the quality of the sample which is actually to be analyzed, the chain of custody and the expertise of the individual who is actually going to analyze it. This means the identification can be shown indirectly through the analysis of traces as well as samples. Again, I'll quote here that no two fingerprints are same. That is, no two parts, no two species or debris or remnants or remains may be same. There will be slightest difference. If you observe it under microscope, there will be slightest difference among the two. Right? So you cannot compare that two. Right? Next comes the law of circumstantial facts. This is mainly concerned with all kind of eyewitnesses who do the testimony, who testify, the victims who give their statements, and so on and so forth. Anytime that people are called upon to provide evidence, there is a chance that the evidence that they supply may not be actually accurate. Every time it will not be as good as they have given it previously. Many times what happens is, if a court is adjourned, the next dates are being given, whatever statement is given by, say for example, victim may not be same. Some of the vital information may be missing. Now many times what happens is that all the testimonies are not actually testified or are not actually recorded every time. What is done is that a large sum or a few important points are being noted, and at the end the final testimony is being done. So many times it happens vital information may be missed out. So it is said that the law of circumstantial facts. This can be unintentional. People actually don't do this. Fine, if I want to say something, if I am in court, if I want to give testimony, if I need to identify a person, if I want to narrate a particular incident, fine, why would I hide it? Fine. So many times we have seen that whatever this they notify the things, fine, so a vital information is being missing out, and that eventually turns the entire case in some or the other favor. It is unintentional through mistake, so mistaken observations and making assumptions which are seldom they are deliberate. In some cases people they lie in court, which is actually a criminal offense as well. Fine. Or what they do is that they talk exaggerating things which are actually not required. On the contrary, we say the evidence which gives a factual account based on the investigation and evidences has a higher chance of being accurate and is considered to be more reliable. So in court it is said that for whatever reason, whatever things that you testify or notify, fine, you should provide evidence for the same. So as per the police record, all those evidence are taken into consideration and the further process is being carried out of the judicial system. Next is the law of probability. Probability, I think majority of you might be knowing, that is occurrence or chance of occurrence. So conclusions that are drawn from any forensic analysis are dependent on the method used, its advantages as well as its disadvantages. Probability and chance of its occurrence of anything is to be considered 100% unless it is out of or totally out of the scope of investigation. Fine, if I found a dead body and I say that whether or not, if I am not seeing any injury marks, if I am not seeing any blood marks, any blood oozing out from the body, how will I am going to say that whether that person has committed suicide or that person was being murdered? Fine, if fixation may be there. Fine, so chances are there that probability occurs in both cases. So this all thing has to be taken into consideration. And if I am finding a dead body in a particular site, how am I going to believe that the crime scene is the same site at which the body is being placed? Chances are there that whatever this murder or anything might have happened at some other place, and the body was later on being thrown out at some other place. It might be transported from one place to another place. Fine. So probability and chance of occurrence cannot be ruled out unless and until it falls in the area or it falls in the investigation screen. Next, now when we talk about forensic biology, it is actually a mixture of many other branches, many small branches. All of these branches have their inputs in forensic biology in one or the other way. I am sure majority of you people will be seeing this and you will be relating it with your subject, that how my subject is applicable in forensic biology, or part of that subject. Entire subject may not be applied, a part of that subject may be applicable. To start off with, we'll say forensic biology, it has got its input from odontology. Next is from the medicine point of view. Third is the entomology. Next is botany. Next is anthropology. We can also have ornithology, then toxicology in it. Then there is a kind of pathology also involved in forensic biology. Microbiology is also involved. Then disease epidemiology, and lastly we say the burning question that hurts all of us now in the modern era is the biological warfare. And we cannot miss out the application of genetics in forensic biology. Genetics has proved to be a very good handy tool in recent past in solving almost all of the paternity and the rape related issues. Now coming on to the first field that has got its application in forensic is the odontology. Forensic odontology, it is a proper handling, examination and evaluation of dental evidences which will be present in the interest of justice. The evidence may be derived from the teeth. We can derive the age in case of children and identification even of a person to whom the teeth may eventually belong. Knowledge of forensic dentistry requires encompassing of number of disciplines. Since the dental records obtained can identify an individual or afford the information needed by the authorities to establish the identification of an individual in the case. There are mainly two areas which are taken into consideration as far as forensic investigation is concerned. First is the investigation of the teeth that is being withdrawn or that is being available to us. Next is comparison of that teeth with whatever bite marks or whatever prints that we have on the body surfaces. That is another issue. So what is happening actually is that at some particular crime scene we may find a teeth. If a dead body is there, a teeth may be withdrawn. Fine, if two dead bodies are there, one is having some bite marks, we may withdraw the teeth of one or the other and we may match. We may actually match the dental pattern, we may actually match the bite marks with it, and that is how we come down to a conclusion that whether or not whatever this teeth is available or whatever bite marks are there are of an individual or are of a suspect that is present or any suspect in that particular case. These are generally taken into consideration wherever there are crimes related to rape, and sometimes it may eventually lead to murder as well. As you can see here, what are the things, the outcomes, possible outcomes of forensic odontology can be? That is determination of species. Fine. If I am there are many cases in recent past wherein in the jungle area or in wildlife sanctuary areas you may find certain nails or certain teeth of animals. Many times it happens that teeth may be of human as well. Fine. So generally if it is canine, it is visible that the teeth may be of a different type, the length of the teeth may be different. But how to identify? Say for example, there are certain endangered species in India. Fine. if that teeth belongs to a particular endangered animal or not, that should be determined. Right? If you obtain a teeth in, say for example, Gir sanctuary, fine, how would you correlate that if it is of lion or a leopard? Because leopards are also available in good number in and around region of Junagadh. So first is forensic odontology helps us a lot with determination of species. You can identify the teeth and to which species it belongs. Next is determination of blood group from that particular teeth. Now that teeth, if it is a molar tooth, you can also have blood from that, and you can do the blood grouping and analyze further that which blood group is there. Then next is DNA analysis. If root is present in the teeth, you can extract DNA from that, and that DNA can be analyzed, and you can establish identity that if these teeth is belonging to which person, whether it is belonging to deceased, whatever a person who has been murdered, or the suspect, anything. Next are the bite marks and the teeth marks comparison. If you see the first and second diagram on the right hand side, fine, the first row of teeth are shown. These are the teeth of a aged individual, fine, an adult individual. Whereas if comparing with the smaller teeth, these are from children. So you can as such determine the age of the deceased person, the person who has died, the person who is being murdered. You can identify if the teeth is present. There is no point in comparing a teeth of an aged person if you have found a child that is dead at that particular crime scene, right? So by that you can determine the age of the dead person. Yeah? Next photograph shows you the bite marks, fine. So how if a person is dead, fine, and on the face, on the neck, at various regions of the body, if you find such bite marks, fine, so you can analyze that what is the dental pattern of that bite marks, fine. You can measure that bite marks, and then later on, if a suspect is there, you can take his or her bite marks as well and compare and superimpose this thing. You can superimpose the measurements, and that is how it is being compared. Next is matching crime scene material with the victim or suspects. Estimation of age, I have already mentioned. Again, you can estimate the sex of an individual, whether that individual is a male or a female. That can also be identified using forensic odontology. Lastly, the facial reconstruction. I will not totally include this thing here because facial reconstruction is something that is also studied when you study the study of bones. Whenever any bones are observed, fine. Facial reconstruction is generally done whenever a heavy any face of an individual is being totally tormented, totally using some heavy stones, or it has been crushed, or if an accident happens, a truck has rolled over, anything can happen and the face is not recognizable, fine. So from the remnants, whatever samples that forensic people give or forensic people have for this analysis, they go for facial reconstruction of that particular individual. If an individual has gone missing and if you find a dead body whose face is totally dormanted, fine, the only option left with us is what we do is from his picture we identify what type of skull bone is there, how it is positioned, what are the different degrees, how it can be rotated, and on that basis whatever the bones we have, we try to superimpose the image, we try to recreate the image, we try to recreate the face of an individual from the skull, fine. So it is a very good technology in case where the person who is or the deceased person who has died cannot be identified on the basis of its face. Next, the forensic medicine and toxicology. Many times, daily rather, in newspaper we might see a news that a few of the entire person of a few families, few members of families, they have consumed certain poisonous chemicals and they have committed suicide. Fine. So chances are there that whatever thing they have consumed, if it is poisonous, fine. If it is consumed, it is entering into the body, it should be found in various body parts. So the entire thing is studied under forensic medicine or forensic toxicology. We'll see it here. The forensic medicine and toxicology, it is a specialized branch wherein the medical science which exclusively deals with dead bodies recovered from the crime scene. I am very sure that majority of you might be knowing common terminology, that is post-mortem. Mortem, that means death, and post-mortem, that is after death. Whatever analysis, whatever studies that are being carried on a dead body, fine, so it is said to be post-mortem. Mortem means death and post-mortem means after the death. There are certain studies which are called anti-mortem, and there are certain studies which are called post-mortem studies. So mainly the forensic medicine and toxicology, it deals with all the post-mortem studies that are taking place. Mostly the forensic medicine department is individually situated in the government hospitals with operation theatre as well as the autopsy facilities. Bodies from crime scene are brought at a forensic medicine division hospital for their autopsy. The doctor or the expert, what he does, he extracts some of the internal body parts. Now the parts that are being extracted or parts which are being cut off are now known as viscera. Fine. If an organ is affected, fine, for visceral analysis, few of the organs are taken into consideration. Likewise the stomach, that is the gut, small intestine, large intestine, a part of liver, part of kidney may be taken for study. So all these are included as viscera. These pieces are then brought to the forensic laboratory for its further analysis. Here the toxicological division takes place, and toxicological division, it comes into play. Whatever analysis is to be done, whether any poison is present or not, this sort of study, the analysis is done and the report is being generated. Viscera brought from forensic medicine division are analyzed chemically for detection and quantification of poison. Fine. Now whatever remnants are present in that particular piece, fine, are then calculated that if this much piece contains this amount of poison, fine, whatever will be the amount that is present in the whole organ and subsequently in the whole body. Fine. So that is how viscera or visceral studies helps us in determining the quantity of poison that has been consumed or any toxic compound that was being consumed. An individual or he or she was forced to consume that. Next is studies related to insects, that is forensic entomology. Forensic entomology is that way important because there are few of the insect species, maggot species, which are eventually growing on the dead bodies. Fine. The time is important in this case. We'll see here, forensic entomology may not be for the faint of heart. I mean, whenever a person who is very weak at heart and if he or she sees a dead body in some distorted manner, maybe if it is a 2-day, 3-day or 4-day-old body, fine, it may be a horrifying scene for us. Fine. Whatever things that we might have seen in movies, fine, that is all makeup that is done and all, but actually to see those type of dead bodies, it is altogether a different scene. Fine. But eventually, if you are strong-hearted, you can see those dead bodies. Fine. But the study of insect on the post-mortem body can yield a wealth of information in criminal cases. Fine. Forensic entomology in a medico-criminal context is often used to, first is, that is to establish the geographical location of death. Fine. The presence of few of the insect species on the body suggests that the scene or the crime scene is the actual scene where the death has taken place and the dead body is not being transported from one place to another. Fine. There are certain species which might be observed, which are the common flora in that particular region, fine, that will be observed on that body. Next is associate the victim and suspect to each other. Fine. To establish a correlation between the two, that is also helpful. Identify the sites of trauma. If you want to identify that at what position this thing has happened, how the body is being positioned, is it the natural position of that body? If a person dies, is it the natural position, or he or she is being implanted in that position? Fine. He or she is being murdered somewhere and is kept and it is being implanted in that particular position. So that can also be studied. Next is determining the time of death. In forensic study, this is the main thing that is being done. That we say it is called time of death or rigor mortis, fine, wherein we judge that if a patient or a dead body is present, we judge how much time before the actual death has occurred. Right? And lastly, It is also useful for providing alternative to toxicology as well as DNA analysis. Forensic entomology, it is a very basic type of analysis that any forensic expert must know. Fine. If he or she is visiting a crime scene, then this sort of study he or she must know, so that to avoid the last stage, that is toxicological studies and DNA samples, because the last thing that is mentioned, these are very costly affairs. Next we will see forensic botany. Plants, medicinal plants, plants have vital compounds, plants have phenolic compounds, plants have all sort of compounds that can be used in one or the other way for betterment, for medicine, and for all purposes for you and mankind. Now, botany in its simplest use is the study of plants. Now, forensic botany is thus defined as the use of plants, plant parts, which may include the pollen, seeds, leaves, flowers, fruits, wood, etc., in the investigation of criminal cases, legal questions, disputes, or in non-criminal cases to ascertain the cause of death or for the location. In general practice, the botanical evidence are not considered directly. They are often called the secondary or supportive evidences. So they support other evidences and generally used for exclusion of innocents and not looking to culprits. Now many times it happens that there is some loot or there is some robbery wherein we'll see that the red chili powder is being thrown onto a person and the money or XYZ thing is being looted. Fine. So in such cases what happens is that this forensic botany is something that can help us. Fine. So what we do is that we analyze the pollen, the flower, and everything that is present in that chili powder. We do the microscopic analysis for that particular case. Fine. So that thing should be present in the crime scene, that should be present in the soil that was present there, that should be present on the clothes, that should be present on the clothes of other both suspect as well as the victim. Fine. So many times it happens that such things are observed and it can help to solve the case eventually. Many times you might have seen that ultimately the robbery was being done by the known person. Fine. So such evidences, they help us a lot. Many times it happens that a person is being falsely being framed into that case. Fine. So these are the evidences, the forensic botanical evidences are the evidences which can help to solve the case in that way that it cannot prove a person culprit, but if he or she is innocent, in that case such evidences may play a vital role. I forgot to mention that there are many other psychotropic drugs and psychotropic plants that are we say that they are being produced by plants or certain vital extract of plants, and they are used as there is an entire section that is dedicated to it, that is NDPS. Fine. So here what is done, that is many drugs are currently into the market. Methadone is there, lysergic acid is there. Fine. There are certain pleasure drugs available. Then there are certain psychotropic agents that are being used, and otherwise very well known are the bhang, charas, ganja. Fine. These are all the substances that are being consumed, that are being grown, and they are being used for various malpractices. Fine. So in that way, study of certain plants, how the growth of that plant looks. Fine. There is a fantastic, if you consider the Indian hemp, or popularly what we call the bhang. Fine. So when they are grown, fine, it attains the position. It is generally grown with the plant that is cultivated. Fine. If it is a wheat plant or a bajri plant, fine, within that wheat or bajri field, a patch of that, along with that, this particular plants are also being grown, and it will attain the same height. It will attain same length, same height along with the wheat and all. But a forensic expert, need to identify whether how that plant looks. Fine. So the characteristic leaves of hemp and all that, these are the things that can help you. If you are coming from a botany background, you should know that what type of venation is that, what type of leaves are present, what is the serrated margin is there, XYZ things. Fine. So that is how botany helps us a lot in identifying. And whenever you are at the actual scene, whenever that lot is being seized, fine, you need to identify whether it is the same plant which is called the Indian hemp or bhang, or any other product that is being derivatized from that. Obviously you will be also given kits for that, we are given the kits for that, for analysis, fine, but still by looking to the plant you should be able to identify that this plant is belonging to a particular category or not. Next is forensic anthropology. Anthropology is study that is related to the skeletal remains observed from a particular crime scene, fine. For any crime scene, if you observe that there are various bones, there are skull, there are skeletal remains that are found. Now this generally happens whenever the dead body is recovered after many days, fine. If a person has gone missing and after many days at some or the other place in some forest area or in some rivers or like that, you may find a dead body, or even if somebody has thrown that dead body or somebody has already done any XYZ activity on that dead body, fine, the deceased person is now not available with us, but whatever skeletal remains are there, fine, now you need to ascertain that these skeletal remains are of that individual who has gone missing. So how are you going to match that thing, fine? Neither the person is available, neither any of his other samples are available, then how to correlate, how to match, how to establish the identity of that particular person? So forensic anthropology, in that way, it is a very good field that is coming handy to us. Forensic anthropology is actually the examination of human skeletal remains for law enforcement agencies, and this helps us with recovery of human remains, determine the identity of any unidentified human remains, interpret trauma, and estimate time since the death. Anthropologists have developed a method to evaluate bones to understand people who lived in the past. Such questions might include what this individual is, that is, he is a male or a female, whatever bones are recovered, whether these bones are of an individual male or an individual female. How old they were when they died, that is, how old they were when they died means after many years the skeletal remains are being recovered. Then if you want to set the identity, if you want to identify who the person was, or whether that person was a male or a female, if you want to establish, and actually what was their age, maybe if a child is being killed at a particular age, fine, and after 4, 5 years or 6 years the case is being reopened. I think majority of you might be well aware about the Talwar's case, fine. And next was the case wherein the Narihari case in Delhi, wherein many child were being sexually abused and they were being killed, and their skeletal remains were thrown, or they were packaged and they were thrown somewhere in the outskirts of Delhi. So how old they were when they died, it can also be established. How tall were they, and what is their height, that can also be established. Were the people in good or poor general health, I mean whether the person was already suffering from a disease and then he has died, or he was being killed and then whatever remnants are there, they are being found, and so the general health of the person who has died, that can also be established. Now how was their stature and overall bodily appearance, fine. As we saw previously that the facial reconstruction, the skull reconstruction helps us to identify whether that individual was the same individual or not, and so that is how from whatever bones and skeletal remains we get, we can identify how the stature of that person was there, whether he was thin, whether he was fat, what was his height, whether he was dwarf, tall, like that. Next is the forensic microbiology and the associated pathology. So forensic microbiology, like other areas of forensic science, deals with determining the cause of death and the identification of people who have committed that crime. Forensic microbiology is a newer area of forensic science and has gained importance after the well-known Bacillus anthracis attacks launched through the US Postal Services in 2001. I hope all of you might have heard about this thing. In 2001, when George W. Bush Jr. he was in the reins of US, he was US president, and this Osama bin Laden, he was being charged for this, that he was behind spreading this Bacillus anthracis in US. What was happening is that a blank label cover would come at your address. Fine, it was being filled with Bacillus anthracis spore, and once you open the cover, there might not be any letter inside that, but what you will do is that you will do like this, and whatever powder is there, it will cover entire or all of your face. Now this powder actually contains the anthrax spores, fine, and these spores are responsible for skin itching and other all the symptoms that are associated with Bacillus anthracis. An important role of forensic microbiology is to determine the microbial signature of an agent recovered in a criminal case. In this regard, the agent's similarity to species or strain is determined in an effort to find the source of that particular agent. Many times it happens that a person is or a sample is being contaminated with microorganism. Fine. So now which microorganism was present, that should be judged. Now what caused that agent to get contamination from microbes, that also needs to be ascertained, and where these type of microorganisms are generally found, that can also be ascertained. Fine. So it is said that if an agent is being recovered, if anything is seen that it is being contaminated with microbiological agents, fine, and we need to ascertain from where do the microbes have entered this particular system, and in general where these type of microbes are being found. So forensic microbiology in a way helps to solve such cases. The phenotypic characteristics have been generally unreliable because they change based on the environmental condition. Therefore, the emphasis is placed on the genotypic variation such as the DNA fingerprints and polymorphism. Forensic pathology mainly deals with the pathological conditions of a dead body at the time of autopsy. Whenever the forensic experts they do the post-mortem studies and autopsy is being carried out, at that particular stage they study all the pathological features as well, whether the organism is whether the disease is suffering from any kind of pathological symptoms or not, that should be ascertained. Next comes the genetics and the DNA analysis. This is the booming field, and I think a majority of persons who are interested in forensic science or are getting fancied by the forensic science is due to the role or due to the excessive use or due to the developing techniques by which the actual DNA of an individual is taking much of the attention in the field. Right? In establishing an individual's identity, first is the fingerprint and next is the DNA. Fine. So DNA is more reliable than the fingerprints as well. Fingerprints can be copied from one individual. Fine. There are many ways. I would not disclose this in an open forum, but there are ways by which a fingerprint of an individual can be copied. But DNA, no, it cannot be copied. If a person is having a DNA, it cannot be copied unless the person or the suspect or the victim is a child of that individual. The part of DNA may match. Fine. Or part of DNA may match if he or she is the sibling, fine, a brother or a sister. So forensic DNA analysis. DNA, I think majority of the participants will be knowing this. That is, it is deoxyribonucleic acid. It is one of the most popular pieces of evidence to recover at a crime scene. Now this is often considered as a golden evidence, as it can easily point to a single person or a pool of suspect. Fine. DNA can never lie. Fine. If a DNA analysis is done, fine, and if a suspect is being proven that he or she has committed the particular crime, it is the utmost truth that one can get. So that is the reason why DNA is considered to be a golden evidence in almost all the cases wherever DNA is involved. However, the DNA collection, the procedure from an older crime scene is more like a headache. Fine. As the samples get old. Now remember the few of the first slide wherein there was a law of change. Fine. So biological samples, as I said, during course of time, all the biological samples they tend to change. they change their physical characteristics, they change their chemical properties. All in all, we can say that the biological material will be degraded or deteriorated. Fine. So if it is a fresh sample, well and good, you can isolate DNA from it. Even you will be getting good amount of DNA from it. But if the samples are older, it becomes very tedious to isolate DNA from that. In many cases, the crime scene is often visited by a number of persons, including the actual investigation team, the police party, as well as the certain local civilians in and around that region who are actually the first who have sighted the scene, or actually who have sighted the dead body at that particular site. In such cases, DNA evidences alone cannot identify the criminal or the victim. Thus, relying only on a single evidence is never acceptable by the court of law. There should be more than one evidences, more than one facts that can prove that a particular suspect is actually committing the crime. DNA can be collected from a number of evidences found at a crime scene. It may be like blood spots or even an entire pool of blood. Any other body fluids which includes saliva, semen, sweat sample, etc. Even a bone can prove a vital evidence in case of DNA analysis. That is, we can isolate DNA from a particular bone. That is, the bone marrow that is present, it can give us a large amount of DNA. Then other thing is the hair. If hair are found with the roots, fine. If that hair is found, only follicles is there, you get very little amount of DNA. But hair along with the roots are present, you may find abundant amount of DNA in that particular thing. So it is very easy to extract it from the hair. In some cases from the nails also you can extract. If there are certain skin remnants are present in the nails, you can remove that thing and you can isolate DNA from them. That may also prove as a golden evidence. Several basic steps that are performed during DNA testing, regardlessly of the type of test that is being done. The general procedure may include: first is the isolation of DNA from the sample that is being collected from the site. Obviously, the origin is not known in this case, and generally at a later time, the isolation of DNA from the sample of an individual of a known thing. And I just want to correlate it. So whatever samples I have found at crime scene, I will try to extract whatever DNA is there, and then if a suspect is there, of an individual is there, I'll ask him to donate his or her own blood. From that blood and other samples, we can isolate the DNA. The processing of that DNA so that the test results may be obtained. Whatever DNA is obtained, it should be in purest form so that analysis becomes easier. Fine. It should not be a mixed sample. The determination of variation in the DNA test results or types from specific regions of DNA. If entire DNA is being studied, you won't get proper results. Chances are there that DNA may be matching, and that DNA, I hope those who are coming from microbiology and biotechnological background, or those who are even working with DNA, they might be well aware that there are various regions in DNA. And you'll be surprised to know that 99% of our DNA matches to the mouse DNA. 99% of our DNA matches to the other higher eukaryotes or other mammalian species. Fine. So proving that DNA as an identity of an individual would be very non-significant. So you need to target various regions present in that particular DNA. Now, as we say, I think many of you might have heard the word DNA fingerprinting. Like as we heard the terminology fingerprinting, there is something called DNA fingerprinting. Now, what is actually DNA fingerprinting? So, in DNA fingerprinting, there are various regions are present in DNA which are occurring in variable numbers. Fine. These are actually a repeat sequences that are present at a regular interval in entire DNA. These repeats are called tandem repeats. Fine. So this may be shorter in size, this may be longer in size. Fine. So if it is shorter in size, we say STR, that is short tandem repeats. So the appearance of these tandem repeats in a particular number, it is the actual identity, or it helps us in establishing individuality of a person. The same number of that particular, if a region is identified, and whatever number that is present of that particular repeats in my DNA may vary from your DNA. It may totally vary. Fine. If it is present in my DNA, it may partly match with my siblings, it may partly match with my parents, but it may totally be different from the other individual. Right? And there are not a single sequence, there are many such sequences that are available, and these variable number tandem repeats, VNTR, variable number tandem repeats, these are taken into consideration, and we say it is establishing an individuality. Right? The comparison and interpretation of test results from the unknown and known samples to determine whether the known individual is not the source of DNA or is included as a possible source of DNA. Fine. So after you, if you have isolated particular DNA from a crime scene, you have got blood, you have got semen sample, you have got sweat sample, you have got skin sample, you have extracted DNA from there, and if a person who is totally nowhere involved in that particular crime scene, he is also being framed into it and he is one of the suspects. Right? So extracting DNA from both individual as well as whatever is recorded from crime scene, and then establishing a comparison between the two. Fine. If it is not at all comparing, then there is no point in conducting such DNA analysis. Fine, because that individual would be innocent, he will be nowhere involved, he is actually framed into that case. So after isolating the DNA from its cells, specific regions are copied with a technique. It is known as polymerase chain reaction, popularly called PCR, and it produces millions of copies of that particular DNA. The resulting PCR products are then separated and detected in order to characterize, as we say, the short tandem repeats, STR. So how the band appears, or what is the number of that band appears, how much that is concentrated, what is the actual amount of that particular sequence or band appearing in the gel. So that is all being studied by electrophoresis or slab gel and capillary electrophoresis, and it is determined that actual what amount or what number or which are the sequences that are present, and such profile is generated for both the samples that are recovered and from the suspect or the victim or XYZ or the person is involved. So there are basically three methods for analysis of specific case. First is the nuclear DNA STR, used for any unknown sample that is collected from crime scene. It gives general idea of the DNA sequence, which contains the mixed DNA pattern and from maternal as well as paternal origin. So it might be helpful in certain cases of rape cases as well as in establishing the paternity related issue after the rape case. Next is the YSTR. This method is basically the STR, but DNA is concentrated and extracted from specifically the Y chromosome, which gives exact match of DNA from the father. So in solving the paternity related issues, the Y chromosome is extracted and the DNA is extracted and the matching is being done. A very recent case, that is of a former minister, it was done, and his son actually, I think he won the case in these regards. So paternity related issues can be solved by this method. Next is the mitochondrial DNA. This method uses extranuclear DNA from mitochondria, which is useful for comparison with the mother. So this method is also useful for degraded material analysis as well as the ancient cells. Future aspects of forensic DNA analysis. DNA microarray. Microarray systems are based on SNPs, that is single nucleotide polymorphism. Any change, DNA is made up of 4 bases, that is adenine, guanine, thymine and cytosine, and any random order of these 4 nucleotide bases makes up the entire DNA, which is obviously many nucleotides long. But when you compare the DNA, then the DNA might be differing from one or the other DNA by a single nucleotide. A single nucleotide, the sequence may be adenine in my case, and it may be guanine or cytosine in your case. So actually this happens. So studying entire DNA by this SNP based technique is done in microarray. It consists of a wholesome analysis system on a microchip like material, and hence it is popularly also called DNA chips. Microarray based forensic DNA analysis and assays, they are capable of, actually they are a very rapid test, Simultaneously, many samples can be analyzed. SNP genotyping for human identification testing for 3 specific forensic samples, that is low template single donor samples, some of the degraded samples, and mixture which is containing more than 2 DNA sources. This 3 type of samples can be obtained from various crime scenes, and DNA microarray based techniques help us in solving cases by all these 3 type of cases. Next is the touch DNA technology, the touch DNA method. Fine. Now, if I touch any surface, whatever cells that are present on my hand, it will directly be placed onto the surface. If I am touching this laptop, if I am touching this table, anything. So, in some cases, wherein there are robbery, where is the murder, fine. So, touch DNA is also, unless you are wearing gloves, fine. If you are touching anything, and who wears now, since COVID scenario is there, people are wearing masks, they are regularly using sanitizer, they are also some I have seen they are regularly wearing gloves, fine. Otherwise it was not a common practice, fine. Unless you are wearing gloves, fine, your DNA is bound to be present on a particular surface that you commonly touch. The touch DNA method, named for the fact that it analyzes skin cells that are left behind when assailants touch victim. If somebody scuffles me, somebody tries to do the affixation, fine. So his hands are on my neck, fine. So many times it happens that his cells might be present on my neck surface. So DNA can be extracted from the cells that are being recovered from this particular surface. So touch DNA doesn't require you to see anything or any blood or semen or any biological samples at all. It only requires 7 to 8 cells from the outermost layer of your skin. Thus it becomes very useful for investigation purpose. And obviously in recent past, touch DNA technology is gaining importance. Next, and the most burning issue, which is obviously the bioterrorism. Some people may relate it to the COVID-19. The SARS-CoV virus, I hope majority of you have known this thing, that there is a virology lab who is doing this all sort of study in Wuhan, China, and there was a theory that this is an outbreak, or purposefully this is a virus that is being sent into the open arena as a biological warfare, as a method to spread bioterrorism, right? But I don't believe that theory, and chances are there that certain epidemic may spread after every 10-20 years. It is bound to happen, fine. But still it cannot be totally avoided. There are certain theories and certain sort of investigations that are taking place, fine. So there is this new word, that is bioterrorism, and how we relate to it and how far it affects us, that should be studied. There is a table given wherein you will see that it is being associated with us since ages, right? It is associated with us since 600 BC. In all of the war, one or the other way, people have tried to use various biological agents, various agents that can cause harm to life, and they are being used in wars, fine. So this is all what is bioterrorism is all about. To kill a person for maybe to grab his land, maybe to grab all his money, or XYZ thing, right? So a biological attack or bioterrorism is intentional release. I forgot to bold this thing, but it is intentional. It is not unintentional. It is definitely an intentional release of any biological product. That is, it may include virus, bacteria, or any other germ that has the capacity to sicken or kill people, livestock, or even destroy crops. Bacillus anthracis, the bacteria that causes anthrax, is most widely and likely agent to be used in biological attack or biological warfare. Up till now it is widely used. An early example takes us back more than 2.5 millennia. Assyrians infected their enemy Wells with a rare ergot fungus which contains a chemical that relates to LSD, that is lysergic acid. What this chemical does, it causes hallucination. It can alter your mental state. So consuming this tainted water produced a confused mental state, hallucination, and in some cases it eventually led to death of the enemy soldiers. In the 1300s, Tartar, that is the Mughal warrior, they besieged the Crimean city of Kaffa. And during this siege, many Tartars died on the hands of plague. So what they had done is that they had purposefully spread the plague in the outskirts of the city area or the walled city. So what happened is that whenever the enemy comes, they will be infected, they will be contracted with these sort of diseases. During World War II, I think majority of us might not be born at that time, but during World War II, many of the parties involved looked into the biological warfare, and with great interest, majority of the allies, they had this facility and they were capable of producing the mass production of anthrax spores was undertaken. Also, it was done for brucellosis and botulism toxin. Thankfully, the World War ended before these things actually were being used as a weapon. Currently, major threats that are being posed to us by this will include anthrax, botulism, Ebola, and other hemorrhagic fever viruses, then plague and smallpox. These are the biological agents that can be used and people can be targeted. So that's it from my side. I am available at this email ID. You can note it down, and this is my contact number. If you have any doubt, any query regarding this topic, you can contact me. I'll be available. I'll be happy to help. Thank you. Happy learning.